All right, boys and girls, this is topic 3.5. This is basically gene technology. Now, this is actually one of my favorite topics, so I hope I do it justice. So basically, you have to think of this as a toolkit. So you have your toolkit, okay? And you have PCR. You have electrophoresis. All right. We have ligase. We have restriction restriction enzymes. All right. We have sequencing. And we have cloning. Now, one by one. This might be a long video. Okay, PCR. Historical. This came from a bacteria called Thermos bacillus. So, extreme bacteria. These bacteria live in deep sea vents, right? So here's the ocean, here's my vent, steam, and these little buggers live right here. So they live at extremely high temperature. Now, why do we care about these little buggers? Well, clearly they have to divide. They have to grow, which means they have to replicate DNA, which means their enzymes operate at very high temperature. As we know, human enzymes like to operate at 37 degrees Celsius. Don't know why I represent. Humans like to live at 37 degrees Celsius, our enzymes. At least, not us, that's way too hot. But we like 37 degrees Celsius for our enzymes. So these bacteria, so what scientists did was they realized, hey, I can use this. So PCR stands for Polymerase chain reaction. And in 2 words, this is a technique to amplify DNA. Oh, there's another one over here. We have plasmids. And I'll explain how these get together. So, okay, look. I'm going to take one step back again. Okay, so PCR, what this does is this will amplify DNA. Okay? This will separate DNA. This will join DNA. This will cut DNA. This will obviously find the sequence. This is copy, and plasmids are a vector. So look, we have a way to select for genes. We have a way to separate genes. We have a way to join them together. We have a way to cut them. We have a way to find the sequence. We have a way to copy them, and we have a way to put them into other organisms. Therefore, we have the complete toolkit to play God. And I don't mean that lightly. We are literally putting genes into different organisms using these different techniques. So if I'm using my enzymes here, I can literally use my restriction enzymes. I can cut the gene out of something. I can copy it using PCR. I can isolate it using electrophoresis. I can join it together with ligase into a plasmid and put it into a bacteria. And then I can take a little bit of that and sequence it and get the complete sequence of a gene. So using all these strategies, I can go from any gene in any organism into any organism. This is how we genetically modify everything. So this is your toolkit, okay? Now, polymerase chain reaction is polymerase chain reaction. Polymerase this is the protein, remember, this is the protein, right? It's in DNA replication. So there's RNA polymerase, DNA, there's RNA polymerase, DNA polymerase, DNA polymerase. What they do is they add nucleotides. So they add ATCG to a growing strand of DNA. So this we have to know a lot of detail about. So the reason why this is important is because this comes from, historically, because we study these extreme bacteria that live on deep sea vents, Thermus bacillus, scientists realized that we could use these proteins to amplify DNA. So the first step is, so DNA is double-stranded, yes? We have a double-stranded DNA. So the first step in polymerase chain reaction is to, one, denature. So we denature the protein. That's the first step. The second step is the annealing. Now see, here's where the piece, so the brand name, I guess you could say like the brand name is TAC polymerase. So this is the name of the enzyme basically. So TAC, when you heat DNA, DNA is going to denature, which means the DNA molecule splits in two. Okay? And so now, the annealing, so now we use primers. Okay, so now the DNA molecule is split. And I have one primer. A primer is about 20 nucleotides long. So a primer is a small piece of DNA. So it's about 20 nucleotides long. So I put a primer on this strand. Remember, my DNA is double-stranded. There's two strands here. I have one primer here. I have one primer here. This is my starting. This is my stopping. So the DNA molecule obviously goes on forever, a lot longer, right? So everything between these two primers is going to be amplified. Okay? So the annealing step is where the primers will attach to the DNA molecule. And so there's like 3 temperatures. So usually denatures at like 95 degrees Celsius. I can't remember where annealing is. And then extending, we lower the temperature, we change the temperature again. And then the third step is the extension. Now the extension, step 3, this is where the polymerase binds to the primer. The DNA molecule comes back together because we lower the temperature. So back here, so now the DNA molecule is back together. The primers are here, and the polymerase is here. So this polymerase goes this way, this goes this way, and bam, we've now copied this bit of DNA. So now we do this. We do steps 1, 2, 3. We do this 30 times. So if you think about it, first I have 1 copy, and then I have 2 copies, and then I have 4 copies. So you do this 30 times, all of a sudden you have millions of copies. And so in your little bottle, in your little tube of DNA, so here's my tube of DNA, and now all of a sudden I have tons of copies of that piece of DNA. So that's PCR. Okay? It wouldn't be possible if we didn't know about Thermus bacillus. It wouldn't be possible because they're the TAC polymerase. This is a DNA polymerase. This is DNA polymerase. This operates at high temperature. Our polymerase, human polymerase, does not operate at human temperature. So we can heat up DNA. We can heat up human DNA or mice DNA, whatever, or bacteria anything that operates at 37 degrees Celsius. We can heat it up. The DNA is gonna denature, come apart. The primers will anneal, which means attach to the DNA molecule, and then we copy everything in between those two things. So now we have millions of copies of that piece of DNA. So that's PCR. Bada bing, bada boom. That's PCR. Okay? Know the 3 steps, know what it does, and know that it amplifies DNA. So our next one is what? Oh, electrophoresis. Okay. This takes explanation too. Now, electrophoresis is basically a way to separate molecules. Okay? Separate molecules by size. Okay? So we're going to focus on gel electrophoresis. Now, there is a northern, a southern, and a western. A western is for protein. A northern is for, a southern is for RNA. Okay, so a western is for protein. A southern is for RNA. And this is where you actually, you know what? To be honest, this is not important. I'm not going to tell you about that. But gel electrophoresis works for a lot of things. What you need to focus on is DNA. So the gel is a protein called agarose. Sugar. So agarose, what it does is it makes a nice gel, like Jell-O. So we have a gel, right? And there's fibers all going through this gel, right? And it's made of agarose. And DNA has to go through all these fibers. So let's say you had a 1% agarose gel. That means 1 gram of agarose and 99 milliliters of water, right? So if I had a 1% gel agarose and I had a 100 base pair piece of DNA, and here's my gel, here's my starting point. If it traveled to here, okay, let's say that's 100 base pair, right? Let's say I take a 50 base pair DNA molecule. It would travel to here. Or I'll color code it. There's my 100. Okay? And if I had a 25 base pair, remember base pair is nucleotides. So I like A, T, C, G, right? So 25 base pair DNA molecule is going to travel the furthest. Okay? Now, what this looks like is, here's my box from the side point of view. Okay? Here's my side. Now, I have a wire here, and I have a wire here. In this chamber, and my gel, here's my gel. Okay? Here's the starting point. Here's the end point. Okay? This is the positive end. This is the negative end. Okay? And in this chamber, we have water. And not just water. In the chamber, we have a buffer. The buffer is basically TBE or TAE. It basically means it's salt. Salt. So this water is full of salt. It makes it a buffer. So now we have a power supply. Here's my DC. Here's my power supply. Power. It comes in here, comes out here. Okay? So the DNA molecule is slightly negative. So DNA molecules are a little bit negative. So when you put your DNA molecule, my DNA molecule goes right here where I start, and I apply voltage. So I apply 100 volts. Here's my 100 volts. It comes in here. It comes out here. It travels through the water because the water has salt. If it was pure water, there would be no current. Okay? So electricity flows through the water, and because the DNA is negative, the DNA gets dragged along through the gel. So if the agarose is thicker, like if it was a 2% agarose gel, or if I had a 3% agarose gel, the lines would travel, the DNA would travel slower because it takes more time for it to meander through. So we can slow down the rate the DNA travels through the gel by increasing the percentage of agarose or by the size of DNA. So what I just did was, okay, so here's my PCR sample, right? I just amplified, here's gene XYZ. I just took this gene and I amplified it, and so now it's in this tube. So now I take this tube and I put this tube into my gel over here. So I put it here. Here's my DNA. And I run it and I apply 100 volts, And so I say I'll do this for 60 minutes. I apply 100 volts. As the current goes through this, the DNA gets dragged along. And then I take this gel, and I put it, there's a few ways to visualize it. So here's my light box, and I put the gel on top, and then this is my side view. And then looking down, this is my bird's eye view. So I'm looking down on the gel. Okay, so now I can see, so like this could be, this right here is my PCR. So if my gene, if the piece of DNA that I was amplifying, if this DNA I was amplifying is only 25 base pairs long, I know it's right there, I can take it out. So what they will like to do on exams is they will basically like show you a gel. So they'll show you a gel, and there will be like 1, 2, 3, 4, 5 lanes. The furthest lane always has a ladder. Now the ladder is basically a known size. How do I know how far? So like this is a known piece. It's already been made. It's been prepared beforehand. So it will be a series of lines, and it's basically a key. So you know this is exponential. So this is going to be like 10,000 base pairs. This will be 5,000 base pairs, 500 base pairs, 100 base pairs. And so you can look at like, so here's a fragment here, here's a fragment here, here's a fragment here, here's a fragment here. So I can say, okay, so this fragment is, it's somewhere around, this fragment is around somewhere around 7,000 base pairs. This fragment, between there, this is going to be like around 700 base pairs. And this fragment is between 500 and 100, so it's somewhere around 300 base pairs. And this fragment is lined up right here, so it's 100 base pairs. Now, this is exponential. So what you would do is you would take a graph, and you would just use a linear equation. So you use, y equals mx plus b. You don't have to actually do this. You'll never be asked to do this. What you'll be asked is you'll be asked to look at a gel, right? So they might give you a gel, and they'll give you. So, but if you know this distance, so if you know the distance of this, and you know this distance for this one, and you measure from the well to the distance, you take the knowns, and you plot the knowns. So now you know the knowns. So now you measure this distance. You measure this distance, you plug it into y equals mx plus b, and you can figure out the exact base pair length of this piece of DNA. Now, you might be given like a picture of a gel, and what they'll do is they'll show like this one be really thick, and this be really faint, this be really thick, and they'll just ask you like, okay, this is sample A, this is sample B, this is sample C, and they're gonna ask you like, so what's the difference? Oh well, look, sample B isn't very much. Now, one thing to know about PCR is that this is quantitative. So, for example, if A, B, and C were different genes, you could say that A and C are activated more than B. Because this is, so if you have a certain gene that's very active in your body, and let's say B is not very active, this could be, it depends on what the question tells you too. This could be mRNA. So we can do electrophoresis. They can give you any picture of a gel, and they can say, oh, this is a protein, or this is mRNA, or this is DNA, right? But that's what electrophoresis is. So electrophoresis is just, it's separating DNA or RNA or protein by size. And we can do RNA, DNA, protein. It works a little differently for protein. This is usually horizontal on a desk. Protein is usually a vertical. So here's your desk, here's it, and they run down this way for protein gels. But, and there's different mediums. It does not have to be agarose, right? Where's agarose? Agarose is a common medium for DNA. You can use, now to visualize it, there's a few ways to visualize it. You can put the gel, so it depends on what you do, right? If you add a certain chemical to the gel, you might use a UV light to see the lines. Or if you use, like if you do an RNA, a southern blot, which is RNA, you're probably using UV radiation. You're using radiation, and you actually put it into a freezer for like a day to expose. It's like film. It's like you put the film on top of the gel, and you put it into a freezer, and a day later you get the results. So there's wide varieties of electrophoresis, but what they might ask you is, what's it for? And this is back to our toolkit. Electrophoresis is to separate molecules. It can separate DNA. It can separate protein. You can look at different sizes. You can know what the size is. And then also, now they're not going to ask you this either, but from the gel. So if you did PCR, you can take your PCR, you can put it into a gel, then you can purify it from the gel. So if I'm just trying to get my one piece of DNA, I put the bacterial DNA into a tube. I can amplify a gene. I can run it on a gel. And on my gel, I can separate the gene I amplified from the rest of the DNA. So here's all the other DNA. Here's my amplified DNA. I can isolate. I can go in there to the gel, cut this piece out, and now I can put this into a tube. And now I have just only the DNA of that gene, and all the other DNA from the organism is now gone. I've removed it from the equation because I separated the DNA on the gel. So it's a way to separate DNA. That's electrophoresis. You might get asked how this works. So there's a gel. What you need to know, there's a gel box. You have your gel box. This buffer in the box is salt. It's salt and water to provide a charge. DNA is electronegative. That's why it runs through the gel. Okay, it runs the same speed. So because DNA is all electronegative, it's all the same. The only thing that makes it harder to go through the gel is how fast it is, how long it goes. So the longer you're always going to see the shorter pieces are going to be faster. The longer pieces, so if this piece is 100 base pairs long, this piece up here is going to be bigger. This might be 1,500 base pairs long. So you always know that the closer to the top is going to be the length of the DNA, the larger length of DNA. Okay, those are the big ones. Okay, ligase, you've learned, this is going to be easy. Ligase, you've learned about this in DNA replication. So basically, what happens is ligase is simply, I'm done actually. Ligase, it's a protein that joins together DNA fragments. So if you think about DNA replication, for HL specifically, you have your leading strand and you have your Okazaki fragments. So these Okazaki fragments are joined together by ligase. So ligase is going to join fragments together. Done. See, that was the easy one. Enzymes. This is important. The fancy word that they're going to use is they're going to call them endonuclease. Endonucleases. These are enzymes. These are molecular, in 2 words, they are molecular scissors. Okay? Now, they have names like BAMH1 or EcoR1. Notice how they're spelled. This means that they are not human. They're not human. Now, historical. What these are bacterial defense mechanisms. That's it. They're the bacterial defense. They're defensive. Okay, so if you are a bacteria and some virus invades you, you're going to let loose your endonuclease to kill the virus. So, because what these guys do is these guys go chop up enzymes. Okay? So they chop up DNA. So, for example, EcoR1 has a specific site. Now I'm making this up. So let's say it's AATT and TTAA. Let's say this is EcoR1's cut site. This is how it cuts. Okay? So every time when you're going along your DNA, here's your DNA molecule. Every time EcoR1 sees AATT, it cuts like this. And it leaves a sticky end. So this piece of DNA now has a sticky end. It has AATT. And this piece of this molecule has the opposite, TTAA. Now, why am I telling you all this? Because I'm going to use a endonuclease and ligase and my PCR product and put a gene into a plasmid. Oh my God, what's going on? Remember, what's a plasmid? A plasmid is a vector. A plasmid is a circular piece of DNA. Okay? Now, so I'm going to do a little story here. Story time. Okay, here's my plasmid. And I'm going to cut it. Now, it has a few cut sites. Now, I have a map. Go Google a plasmid map. Just Google plasmid map right now on the screen somewhere. And you're going to see a circle with a bunch of cut sites. And this is EcoR1. And this is BAM-H1. And this is behind 3. These are all real enzymes. Google them. You're going to find them. You'll find the cut site. You can tell me what I'm doing wrong. Okay? So what I do is I take this plasmid and I take these enzymes and I cut it. So if I cut this plasmid with EcoR1, I now have a linear piece of DNA. And on this end, I have AATT, and this end is TTAA. Now I take my PCR product. Here's my PCR amplified gene. Now what I do is I cut that baby with the exact same one. So I cut this guy also with EcoR1. So now I have a piece of DNA that also has AATT. Now I take both these pieces together. I put these together with what enzyme? What enzyme, people? Ligase, right? There's my ligase. So now I use my ligase, and I have bada bing, bada boom. I now have this plasmid, and I have my PCR product in the middle. Okay, so a 3-step process, right? The restriction enzymes cut the DNA, right? And then I add the PCR with the plasmid, and I join together with ligase. Those are my 3 steps. 1, 2, 3. Now, the reason why I have this map with multiple sites is because I don't know which way it is. Is this the correct direction? Is the gene in the correct direction? So if I knew exactly where I want to cut it, I can cut this plasmid with EcoR1 and BAM-H1, and I take my PCR product and I cut it the same way. So now I know I want this side to be front and this side, I want this to be first. I want this to be last. So now I can make sure that I can put in my PCR now. So let's just call it a gene. This is back to my fancy name, gene XYZ. Okay? So, what do you need to know? Endonuclease. These are proteins. They're enzymes. And they cut DNA at specific locations. They are protein, sorry, I get messy here. Protein enzymes, they're molecular scissors. They come from bacteria. They're defense mechanisms against they're a viral defense mechanism in bacteria. And when they cut, they leave sticky ends. That is actually go Google it. I'm not kidding you. This is actually what we call them in a lab. We refer to them as sticky ends because this piece of molecule, this DNA molecule, this double-stranded DNA molecule now has a single-stranded sticky end, and it can join. This molecule is this DNA is still double-stranded, right? So it's just the end is now sticky because it's single-stranded on the end. So we can organize and orchestrate where we want pieces. So enzymes are used to cut DNA. Okay. Now, oh my God. Okay, cloning. I'm gonna go to cloning now. Cloning. Now, see what I can do is I can take this so now I have my plasmid, right? And I have my PCR product. I have my gene in the plasmid. Now I can take this baby and put it into bacteria. And the process by which we call that is called transformation. So transformation is the process of taking a bacteria to absorb this plasmid. Now we do this with eukaryote cells. We do it with cancer cell lines. This is called tissue in a lab. We call this tissue culture. So we can culture bacteria or even cancer cells or stem cells. We can. This is how we make This is how you make genetically modified mice. Okay, so what happened is that if you look go Google a plasmid map. Usually on a plasmid, you're going to have maybe 3 to 5 genes. And one of those genes on there is basically going to be an antibiotic. So this gene makes the plasmid resistant to antibiotics. So what I can do is there's 2 ways you can redo it. There's 2 ways. You can heat shock. You can take your bacteria and you heat shock it, or you shock it with like calcium. So you either do and when I did this in a lab, we always did heat shock. So we heat up the bacteria real fast. It makes the membrane permeable, and the plasmids are absorbed into the bacteria, and now the bacteria has a plasmid. And then you put it on a petri dish. You add the petri dish with antibiotic, so that all the bacteria on the plate are going to die except for the ones that have the plasmid. And the plasmid has my gene in it. So now I've successfully made bacteria with my gene. This is, who's heard of insulin? Of course you all have insulin, right? For diabetics, we make insulin this way. So one of the very first genetically modified bacteria was they made bacteria that can make insulin. And this is how they made insulin for people who are diabetic starting in the 80s and 90s. So this is one of the first times we actually used plasmids to make drugs. This is very common, extremely common. If you do biology in university, this will be the first lab you do. It's standard practice, right? So and in the 90s, this shocked the world, like literally. There's a conference going on, and some student was like, "Hey, can I do this?" And people were like, "No, you're playing God. We can't do that. Let's put a moratorium." And all research was stopped for a couple years as they debated what should be the rules about this. Now we have CRISPR, and CRISPR is the exact same thing. CRISPR, which we're not going to get into because that's not going to be on the exam yet anytime soon, but CRISPR is another bacterial defense mechanism, and this allows us to edit living cells. So, when you put a gene into a cell, then you have to grow it up, and you're not making a whole new organism. With CRISPR, I could actually give you a drug, and it can modify your cells currently right now. So if you were sick because of some mutation, I can go in there and fix your DNA. In theory, this has not been done yet. It's much more complex than that, but that's the prevailing hope with CRISPR. Scientists in China have already modified human embryos with CRISPR, but they modified embryos that could not survive being put into a surrogate mother to be born. So they took non-viable embryos and they modified them with CRISPR. So in theory, we could fix any genetic disease that plagues humans. That's a problem for humans. Okay, so we're almost done. Oh my God, 33 minutes. That's plasmids, cloning, sequencing, enzymes. Actually, I could probably do a whole video on just how to do cloning. Go through the steps, because basically we use all these steps for cloning. So if I'm doing a clone, right? If I'm cloning, I'm gonna amplify the DNA using PCR. I'm gonna isolate. This is also isolate too. So I'm gonna amplify the gene that I want. I'm gonna separate it and isolate it using electrophoresis. I'm gonna take my endonuclease and I'm gonna take ligase and I'm gonna cut both the PCR and the plasmid, and I'm gonna put them together. And then I can use transformation to put that plasmid into a bacteria, and we call that cloning. So I've now used all of these steps except for sequencing to do cloning. That's bacterial cloning. The other kind of cloning, there's two kinds of cloning. Okay, so back to the syllabus. So, okay, so cloning. There's cloning where it's called bacterial. We use bacterial transformation, and basically what we're doing is we're adding a plasmid. We're adding a plasmid with a gene of interest, and we're going to add it to a bacteria, right? And the good example for you to Google and learn about would be insulin, would be a good example of how we've done that. The other kind of cloning is somatic cell nuclear transfer. So this was the famous sheep Dolly. Dolly is a sheep, and what they did was they took here's my nucleus of one cell, and here's my, so here's my other nucleus. This looks very inappropriate, but bear with me. Okay, so we take this nucleus and get rid of it. And we take this nucleus and put it into this cell. And then we take this cell and put it into a pregnant sheep. Boom. I mean, that's horrible drawing. That's all this is. It's somatic cell nuclear transfer. You take the nucleus of one cell and put it into the egg of another cell. It does not look right to me. It just looks inappropriate. So here's my egg, right? So my egg from my ovary, right? I remove the nucleus and put it in the nucleus of one of your cells, and you got a brand new baby. And they've done it for sheep, and that's kind of it. It's not very common, and I don't think there's a lot of, I don't know how much actual research is done these days on this. I did see an article recently about some lizard or something. I don't know. I think the hotbed of research right now is CRISPR. People are all the rage about CRISPR. It's huge. CRISPR. Okay? It actually might come on the exam. It might come on the exam in some weird way, but they wouldn't be asking you what it is. It's not in the syllabus. So they won't be asking you, like, they might ask you about endonuclease. They might ask you about electrophoresis. They might ask you about PCR. They're not going to ask you about details of CRISPR. So there's no point in going over details unless you really want it. Okay, sequencing. Oh my God, we're almost done. Sequencing. This is a pain in the ass as well. Oopsie, sorry, you didn't hear me say that. Okay, sequencing. Okay, so you need to know what DTNTPS is. Okay? DDNTPS. Where is it? Okay. Dideonucleotides. Now, what these guys do is they stop replication. So so imagine this way. So here's my DNA molecule, right? And it goes A, T, C, A, T, G, G, C, A, T, T, G, A, C. Okay, so basically you're copying this DNA over and over and over again. Every time, and now so DNTPS stands for nucleotides. So we have ATP, CTP, GTP, and TTP, right? ATCG. Okay? So when you add the DDNTPS, this stops replication. So what happens is, so here's my test tube. So I have my test tube, right? And now I have a fragment that's just A. And then a DDNTP gets incorporated here. So now I have AT. And now I have ATG. And now I have ATGG. Now I have ATGGC. So now so that's for the whole entire strand. Okay? That's my piece of DNA I'm trying to sequence. So now I have all these different fragments floating around in my test tube. So when I put this into a gel, what's going to happen? Which piece is going is going to run the fastest. So this A is going to be down here, right? And then the next piece is going to be like a little bit further up. So this is AT. And so what you're going to get is you're going to get like ATC, ATGC. So this A will be down here. And then you have AT. Then you have ATG. So that would be here. Then you have ATG would be here. And then ATGCC. So as I go up, so my next fragment would be ATGGCA. And my next fragment would be ATGGACAT. And then T again. And then G. And then A. And then C. So I get this gel. So here's my electrophoresis gel. So now I have all these fragments. That's basically how sequencing works. Another way they do it, and then that they have is kind of have like a light scanner. So there's like, so as this, it gets confusing. Okay. This is, okay, we're using PCR here basically. We're amplifying a piece of DNA. So we're using primers, right? So if I'm sequencing, so let's say here's my piece of DNA molecule, and I want to sequence everything between these two pieces, right? So what I do is so here's my DNA molecule, and I want to sequence, but I want to sequence here. So I pick a primer here, and I pick a primer here. So I'm going to PCR everything between before and after the part I want to sequence, okay? Now, in practice, this is about 5,000 base pairs long, max, okay? So, and then I add, so but differences in PCR, I would just use DNTPS, right? Because I want to copy the whole piece of DNA. So I just add A, T, C, and G. I add ATPs, TTPs, CT. So that's DNTPS. So that's normal PCR. But in sequencing, I want to make fragments. So from this point to from point A to point B, I'm going to make fragments of every little piece, right? So I'm going to use DDNTPs. So now, instead of having 30,000 copies of the same, I have all these different fragments. I have all these different fragments of different sizes, everything from point A to point B. And so now I take this and I put it through a machine. And in this machine there's a little like light sensor here too. There's a little like light, I think it's a laser, or something. There's some kind of sensor here. So as this test tube comes through here, it scans it. So, okay, so it goes it gets this. So each fragment has different size. And then, so what you get is you get this chromatogram. So you get this like, there's A, T, C, and I'm going to actually download a picture. So I'm going to pause for a second. Woo, I'm back, look at me. Okay, so look, so here's my chromatogram. So this is what I'm talking about. So here is what I'm talking about, right? So look, you have each piece. So this, this is A, T, A, and you can see these little peaks right here, right? So when you send off your DNA sample to the lab, so when I did this in the lab, I didn't actually do sequencing. I would just, when I wanted to sequence something in the lab, when I wanted to sequence something, I would just order primers. So I would order a primer, and I would send off my sample of my DNA to the sequencing lab, and I will give them my primers, and they will make the primers, and they will send me back this. So there's my, this is the base pair, so that's 10 base pairs. T, G, T, G, and so you can see the peaks. So G, T, G, T, G, A, 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 T, T, G, T, T, A, T, and so on, right? So there's my chromatogram. This is how it looks on a gel, so you can see that. So so basically, they've given pictures on old exams, they given a picture of the gel. They gave a picture of just this. And you had to figure out the sequence. So you got to know, you got to go up. And the reason why it works is because of DDNTPs, diiodeonucleotides. So it's like remember, I think previously, oh no, not, so remember, when I was doing my last video with the respiration, right, I talked about ATP, right, ATP, right, so adenosine triphosphate, there's my nitrogen base, and there's my phosphate group, right? So when I say DTNTPs, I'm talking about ATP, GTP, TTP, CTP, because there's 4 bases, A, T, C, G. Okay. Wow, look at that. Look what we did. Oh my God, 45 minutes long. And that's it. That's sequencing. That's a little bit of CRISPR. I'll give you some side notes. So what's cool is that in the human genome, remember the Human Genome Project? You gotta know what this is too. The Human Genome Project, I think this is 1.5, okay? The Human Genome Project. So there was, okay, so here's my DNA molecule. Right, here's my chromosome. So what scientists would do is, because they could do 5,000 base pairs at a time, the government was going, I'm going to sequence this, and then I'm going to sequence this, and then sequence this. And they kept just sequencing 5,000 at a time. So if you imagine you're trying to do 46 chromosomes, and you're doing that 5,000 at a time, it's going to take forever. Oh my God, it's going to take forever. This is why the Human Genome Project took 5 years and cost $1 million to do the Human Genome Project. I just listened on a podcast, not kidding. Last week I heard a podcast. It's down to $1,000 now. $1,000 I can sequence your genome. 5 years ago, it was $5,000. Okay? When I started teaching 14 years ago, it was still like $30,000 to do a human genome. So it is incredibly cheap now to do a human genome. Now what's cool though is they did this with, I think, fruit flies or something. So with fruit flies, this company that was competing with the Human Genome Project did a whole different method. And what they did was they used restriction enzymes, endonuclease. So instead of just going bit by bit by bit by bit and going down the chromosome, what they did was here's my chromosome, they took my restriction enzymes, and they just chopped this up. They chopped it up into 1 billion pieces. And then they sequenced all those, they just sequenced the whole thing. But then, what did they use? They used the magical computer. And basically what they did was they just basically cut up the chromosome into many random pieces, sequence the pieces, and then use the computer to line up all the pieces. So you only need, honestly, the primers, like I keep talking about, the primers in PCR and sequencing, they're only 20 base pairs long. If I came up with a 20 base pair sequence, it might only appear once or twice in your entire genome. So you don't need like a lot of base, you don't need a huge primer to get very specific. You can, with 10, 15 base pair long, you can hone in on a specific spot on a chromosome, because that sequence is going to be unique and nowhere else in the entire genome. So this company was competing with the Human Genome Project, and eventually they lost because the Human Genome Project won, but the Human Genome Project ended up adopting their strategy because it was faster. And that, going all the way back to the beginning, that's our toolkit. If you need help, you let me know.